Okay, so welcome to this next video in which we are discussing the mammalian circadian timing system. Okay, right, so we've now discussed that every cell within the human body has this intrinsic circadian oscillator, which has this ability to allow the cell to know what time it is. Now, the problem is that all of these clocks are bound to get out of synchrony, basically, if you just let it go. Okay, so there is a master clock. There's two of them, in fact. There's two suprachiasmatic nuclei, which are up in the hypothalamus within the brain. Okay, and these are all oscillating in sync with one another. So it's a collection of neurons, and all of these neurons are oscillating in sync with each other. And moreover, they're oscillating in sync with the light-dark cycles of the external world. And these then send messages, which seem to be diffusible molecules, out to all the other tissues of the body, to all the other cells, to tell them what phase their circadian oscillator should be at, basically, so that all the circadian oscillators in all of the cells of the body are oscillating all in sync with one another. Okay, so what we want to look at now is the pathway by which the neurons of these suprachiasmatic nuclei become in sync with the light-dark cycle. Okay, so for this we're going to have to start with the eye, because the eye is where we get visual information. So visual information is going to be coming from the retina to the suprachiasmatic nuclei, and we want to look at how this works. Okay, so we're going to start off then with the anatomy of the eye, the gross anatomy of the eye, just to make sure everyone's familiar with that. We'll then go to the in-detailed anatomy of the retina, so the different types of cells within the retina, and we'll then look at the cells that are within the retina that are going to be communicating information about the status of the light-dark cycle to the suprachiasmatic nuclei. Okay, so the gross anatomy of the eye then. Okay, so whenever drawing a picture of the eye, it's a good idea to start by drawing a circle. Okay, so we'll start with the outermost layer, which is the sclera. Okay, like so. And then at the front, the sclera sort of bulges out and forms the cornea. Oops, that's overshot a little bit. Okay, and of course it's not going to be it's not going to have no thickness, so we'll give it a thickness here. Okay, so this is the sclera coming back like so. And then the sclera is also going to cover the optic nerve that is going to come out at the back here. So we'll have this extension of the sclera backwards like so. But we'll bring this round first like so. And then this too will extend backwards to coat the optic nerve that's going to run in here. So all of this is supposed to represent the sclera. Okay, so the portion that surrounds most of the eye is called the sclera. Okay, and it's the white of the eye. So when you look at the white portion of the eye, that's the sclera. Right at the front, where it covers the iris, it moves out, it protrudes a little bit, and this portion is called the cornea. Okay, right. So the next layer inwards, so if we're going inwards from the outside, the next layer is going to be a layer called the choroid. Okay, now the choroid does not coat the optic nerve, it starts here. Okay, and we'll colour in all of these layers later. Now, as the choroid moves forward, it's going to produce the ciliary body here, and it's also going to be continuous with the iris, which is going to be here. Okay, right, and now we just need to draw the opposite, well, it's on the opposite side as well. So here, this is the choroid coming forward like so, and I want to get the ciliary body at the same point. So here's the ciliary body, and then the iris once again moves forward to have a gap in between them, which is known as the pupil. Okay, right, so we'll colour in the choroid then in red, and I shouldn't have coloured the iris in red, but never mind. Not many people have red irises, never mind though. Okay, so this is the choroid coming all the way back here, and as I say, it does not cover the optic nerve, so it stops short of the point where the optic nerve is going to come out of the eye. Yes, this is the choroid here, and then we've got the ciliary body coming forward here, and then the iris coming forward here as well. Okay, so These two pieces of muscle here, this and this, these are the iris. Well, these are the... Because we're only seeing a slice, of course, we can only see parts of the iris. In reality, the iris is an annulus that surrounds this circle in the middle called the pupil. Okay, so here is the pupil. Now I've drawn the pupil arrow where I want to later put the lens, but never mind, I'll extend it back here. Okay, pupil. So we haven't completed this structure yet. So we're going to have the suspensory ligament emanating from the ciliary body, and then sitting here we'll have the lens here. Okay, so this is the lens which focuses the light onto the retina. Okay, and it's suspended from the ciliary body by the suspensory ligaments here. Okay, right. And before I put any more arrows on, which are going to distort the picture, let's firstly finish the picture. So we now need the retina here. Okay, so the retina starts quite near the front. It doesn't go all the way around, obviously, because no light's going to get to here. But it comes around like so. And then it's continuous with the optic nerve. Okay, so it's going to send in neural tissue into the optic nerve here. Okay, now we want the same in principle on the other side, but I'm going to struggle to put the same thing on the opposite side because I put these labels here. Okay, but never mind. Right, so let's now colour in the retina in yellow. So here we have the retina going all the way around here. Okay, and then it's continuous with the neural tissue that's then going into the optic nerve, because all the neurons from the retina then go into the optic nerve here, like so. And we would want the same thing on the other side, but I think I'm going to smudge that label if I try and colour it in, but I'll have a go. Okay, so here's the retina on this opposite side of the eye here. Right, and then we've got the optic nerve neural tissue here. So this is the optic nerve. Okay, whoops, optic nerve. And then we've got the retina over here. Right, so what other structures do we need to put on here? Well, we're nearly complete, in fact. In fact, I think we are complete. So I won't colour in the sclera, because obviously the sclera is white. Okay, there's just a few more... Oh, there are some more structures I want to put on here. I want to put in the artery and the vein, which supply and drain blood from the retina. Okay, and these move up through the middle of the optic nerve. Oops, which is why I've left that gap down the middle. Okay, so this artery and this vein here, those are called the central artery and the central vein of the retina. So we have here, moving up through the optic nerve, the central artery and vein of the retina. Okay, so those are just a few extra little structures that I wanted to add on. Okay, so let's now discuss the chambers of the eye. So this large chamber that you have behind the lens and in front of the retina, this is known as the vitreous chamber. Okay, and the vitreous chamber is full of a gloop, basically, called the vitreous humour. If you've ever done dissection of the eye, you will know what I'm talking about. It's basically a large sort of jelly-like gloop that sort of flops out, and you can't believe it comes out in the, when you first see it. But that's what fills this vitreous chamber, so it's not just a void of air, it does have a quite viscous jelly within it. Okay, then we have these two chambers here. Okay, so at the front here, it's difficult to put arrows on those, but I'll have a go. So this chamber here, which I'll describe to you, is behind the iris, okay, but in front of the lens and the suspensory ligaments. This is known as the posterior chamber. Okay, so do not get confused and think that the posterior chamber means the vitreous chamber. Okay, even though the vitreous chamber is more posterior than the posterior chamber is posterior to the iris. Okay, this chamber then that's in front of the iris is then, and behind the cornea here, this is called the anterior chamber. Okay, because it's anterior to the iris. Okay, and in both the anterior chamber and the posterior chamber, you have something known as aqueous humour. Okay, so the posterior and the anterior chamber are in connection with one another via the pupil, so they contain the same fluid. And this aqueous humour is secreted by the ciliary body, which is this structure here. Okay, so that's called the ciliary body. The ciliary body has both glandular function and also muscular function. It can contract and control the topology of the lens and therefore affect the focusing of light onto the retina. Okay, but in addition, it also secretes the aqueous humour into the posterior chamber, and then the aqueous humour moves from the posterior chamber into the anterior chamber. Okay, right. So that's the gross anatomy of the eye. Now what we're going to focus in on is this retina, okay, because that's where our photoreceptive cells that are going to be sending signals to the suprachiasmatic nuclei about the state of the light-dark cycle are going to be present. Okay, so what we're going to do is we're going to have a look at the microanatomy or the histology of the retina. And we're going to start off from the bottom of the retina, so from the portion that faces the choroid underneath. Okay, and did I ever label up the choroid on this diagram? I don't think I did. This is called the choroid, this layer between the sclera and the retina. Okay, we're going to start off with the portion of the retina that faces the choroid, and then we're going to go medially, basically, or towards the center of the eye. We're going to go in towards where the vitreous humour is. Okay, so let's do this down here. So, the bottommost portion then of the retina that faces onto the choroid consists of a layer of cells that are known as the retinal pigment epithelium. Okay, so I'll draw these like so. Okay, so these are called the retinal pigment epithelium, and the retinal pigment epithelium is extremely important. It is involved in the recycling of the retinal pigment, okay, after light has hit it. So after light hits the retinal pigment, which is 11-cis-retinol, it changes to all-trans retinol, okay, and after that it has to go through a regeneration process to return it to 11-cis-retinol, and a good portion of this is carried out within the retinal pigment epithelium. Okay, so this is extremely important for the recycling of the visual pigment, and without it you would very quickly run out of retinal pigment and no longer be able to see. Okay, so the retinal pigment epithelium is abbreviated to the RPE, and I've just realised how little space I've actually left here. We'll have to have a good go at squashing this all in to this small amount of space. Okay, then sitting on top of the retinal pigment epithelium, you now have the photoreceptors themselves. Okay, now the main two types of photoreceptors within the eye are going to be in this next layer. Our type of photoreceptor that we're interested in, the type that is actually going to be sending information to the suprachiasmatic nuclei, that is not going to be in this layer. Okay, but the two most famous photoreceptors are in this layer. Okay, and the two most famous types of photoreceptors are rod cells and cone cells. Okay, so let's colour this cell in here. So this portion that I've got down here, coloured in purple, this is the rod of a rod cell. Okay, so the whole cell is a rod cell, and the rod cell has a cell body here, and it has two processes coming off it. Okay, one is an axon which will go and synapse to the next neuron along, and the other leads to the portion that is actually responsible for phototransduction. Okay, and this, in the case of rod cells, is actually in the shape of a rod here. Okay, so this is the portion where light will actually be transduced into an electrical stimulus, basically, or an electrical signal. Okay, right, let's draw the other major type of photoreceptive cell then, which is a cone cell. Okay, and cone cells, the portion that they use to transduce light into an electrochemical signal is in the shape of a cone, which is why they're called cone cells. So this portion that I've highlighted in green, this is the portion which will actually transduce the light into a signal within the cell. Okay, right. Now what's going to happen is these two photoreceptors are going to transfer their message, their signal, onto the next neuron along. But before we discuss that, I want to discuss a membrane that you have that is sitting here between the photoreceptor layer down here. So this portion which contains the actual rods and the actual cones of the rod cells and the cone cells, this is known as the photoreceptor layer of the retina, and it's sitting just on top of the retinal pigment epithelium. Okay, and people always find this odd when they first learn about it, because the retinal pigment epithelium, remember I told you we were starting right from the base of the retina, we were starting from the choroid. Okay, we are starting from this side here. So, right on top of the retinal pigment epithelium, which is the bottommost layer of the retina, you then have the photoreceptors themselves, and they are underneath a huge pile of rubbish that I haven't shown you yet. So the light is going to have to come through all of this rubbish to get down to the actual photoreceptors, and people find that surprising at first. I found it surprising. Okay, I still find it surprising. Why wouldn't you have the photoreceptors on the surface where the image is nice and clean? Okay, but that's the way it is. So this is the photoreceptor layer right down here. Then separating the actual photoreceptors from the cell bodies of the rod and cone cells, there is a membrane, okay? And this membrane is known as the external limiting membrane. Okay, and later what we'll see is that there is an internal limiting membrane as well. Now this one is the external one because this is most peripheral, okay, so it's towards the outside of the eye. Remember, we are working from the outside and going in, so that's why we've met the external limiting membrane before we've actually met the internal limiting membrane. Okay, now, sitting just on top of the external limiting membrane, you're going to have a layer of cell bodies, which will be the cell bodies of the photoreceptors, so the cell bodies of the rod cells and the cone cells. Okay, this layer of cell bodies is known as the outer nuclear layer. Okay, again, it's the outer nuclear layer because there's going to be an inner nuclear layer, and we are working from the outside towards the inside. Okay, so let's now discuss the next neuron along in the photoreceptor pathway. Okay, and these are the classical photoreceptors. These ones are going to be sending signals to the primary visual cortex eventually. Okay, through a quite complicated pathway, but still. They're not going to be sending information to the suprachiasmatic nuclei. They're far too important for that. There's going to be other ones that do that. Okay, right. The next neuron along then is a neuron known as a bipolar neuron. And again, it's having to be shown quite small because of space constraints. Okay, so the bipolar neuron is called a bipolar neuron because it has two processes. Okay, so it has two poles, if you like. So it has these two processes coming off it. One which is going to synapse with the axon of the photoreceptors, so of the rod cells and the cone cells. Okay, so this one here is synapsing with the axons of the rod cells and the cone cells. Okay, these two here. And then they have another process which is then going to transfer the electrical signal onto the next neuron along, which will be the ganglion cells. Okay, right. Now, this layer then of the retina, which consists of axons of both the photoreceptive cells and also of these bipolar neurons, this is known as the outer plexiform layer. Okay, again, it's the outer plexiform layer, and there's going to be an inner plexiform layer because we are working from the outside towards the inside. Okay, so this is the outer plexiform layer. Then this next layer of cell bodies that you have here, which is the cell bodies of all of the bipolar neurons, this is called now the inner nuclear layer. So we've seen the outer nuclear layer, and now towards the inside, we've got this inner nuclear layer, which consists of the cell bodies of the bipolar neurons. Okay, so after the bipolar neurons, what you then have is ganglion cells. Okay, so here are the axons of the ganglion cells, which are going to synapse with the axons from the bipolar neurons. Okay, here are the cell bodies of the ganglion cells, and then they're going to send their fibers up into a layer above the cell bodies, which will then move to the optic nerve. So these fibers will be running in the outermost layer of the retina towards the optic nerve, and then they'll go into the optic nerve. Okay, and then on top of this layer of ganglion cell fibers, you then have another limiting membrane, okay, which stops the nerve fibers coming in contact with the vitreous humor. Okay, so this membrane now is called the internal limiting membrane. Okay, right, because it's the most internal of the two limiting membranes. And as I say, it will stop the nerve fibers of the ganglion cells from being in contact with the vitreous humor. Okay, so outside of this, we then have the vitreous humor. Right, so let's label up some more layers now. So this layer here, consisting of axons of the bipolar neurons and also axons of the ganglion cells, this is known as the inner plexiform layer. Okay, so we have the outer plexiform layer, which consisted of axons of the bipolar neurons and axons of the photoreceptors, and now closer to the inner aspect of the eye, we have the inner plexiform layer. Okay, right. You then have another layer of cell bodies, which is known as the retinal ganglion cell layer. Okay, and for short, retinal ganglion cell is abbreviated to RGC. Okay, so R stands for retinal, G stands for ganglion cell. Okay, so this is the ganglion cell layer, or if you want to be utterly precise, you can say retinal ganglion cell layer. The layer then of nerve fibers of the ganglion cells is known as the fiber layer. Okay, right. So this is the structure then of the retina. These are the pathways which will lead to visual information going, as I say, to the primary visual cortex, which is the normal mechanism by which you see. However, there are other cells other than rod cells and cone cells that are within the retina which themselves are receptive to light. Okay, and these are actually ganglion cells. So you have in the retina certain special ganglion cells. Okay, drawn here, like so. So I'm going to highlight this retinal ganglion cell in a special colour. So this one in orange is not a normal retinal ganglion cell. It is not getting information from rod cells and cone cells. Instead, it gets visual information on its own accord, basically. Okay, so this is what's known as a PRGC. Okay, and what does PRGC stand for? It stands for photoreceptive. That's what the P is for. Okay, photoreceptive, and then the RGC then stands for retinal ganglion cell, as before. Okay, right. Now, these photoreceptive retinal ganglion cells are actually capable of sensing light for themselves. They have within them their own retinal pigment, okay, or photoreceptive pigment called melanopsin, which is completely different from the pigments that you have in rod cells and cone cells. In rod cells you have rhodopsin, in cone cells you have iodopsin. So in these photoreceptive retinal ganglion cells you have melanopsin. When light interacts with melanopsin, it will trigger pathways which lead to the ganglion cell firing action potentials. Okay, the axon then of the ganglion cell is going to go into the nerve fiber layer, and this then is going to go into the optic nerve, and it's going to go towards the suprachiasmatic nuclei. Okay, right. So these photoreceptive ganglion cells then send their axons to the suprachiasmatic nuclei. Okay, so let's draw again the picture of the optic chiasm. Okay, so we have here the optic chiasm with the two optic nerves coming in and then the two optic tracts coming out. Okay, so here is the left optic nerve here and the right optic nerve here, and let's say, well, we're going to have these axons of these photoreceptive ganglion cells coming into both optic nerves, and they're then going to go to the suprachiasmatic nuclei. Okay, and we'll continue this discussion in the next video.